My name is Dr. Antonor Athrell Hinton Jr. Most people call me HA. The goal of our research is really to understand basic mechanisms, meaning how proteins interact in the cell, kind of like if you're having a NASCAR track and they're colliding with each other. That's kind of what's happening in the body. And so that's what we're studying, and we're trying to actually calm that down so that we can actually focus on just watching the rays and not watching them crashing into each other. We're using FibSim as a diagnostic tool to really be able to look at mitochondrial structure, and the software plays a key role in that.
When we do all these imaging in cerebrock facing SEM or a FibSim, you have this tissue, you're going to take lots of pictures, and they're all serial sections of each other. And you're going to get hundreds of pictures. You're going to have mitochondria, you're going to have the nucleus, you're going to have all these different organelles. But what we want to do is that we take all these images, 200 to 300 images, and we put this into Amira. What we do in Amira is that we really want to get rid of all the noise that's present. And so when you get rid of all that stuff, you really just have what you really want to look at.
If I circle the wrong mitochondria or circle a lipid droplet instead of a mitochondrion, I can actually go back and fix that, which is helpful for me because I can visualize it every single time. And so that's also makes it easy to train and learn and teach students with, because you can say, oh, well, actually this is what this is, or let me show you how to fix this. They can actually see it in real time.
It is still simple enough that we can have our undergraduate students, and we even have sometimes high school students come in, and we can teach them how to use the program, and then they can play an integral role in the science that we do. It has the ability to take the structures that we make and export files for 3D printing. So we can take these real-life, real-world examples of these cellular organelles, cellular components, and print them and use those as teaching tools in our outreaches.
It's so beautiful because we can actually look at these things in 3D now. Who knew that mitochondria had more than one form and function, right? Because 20 years ago, if I was to tell you that there was 8 different mitochondria and how they look in their structure, you would have told me I was crazy. Now I can show you that I have evidence that this is happening.